Welcome back to the lecture series in role of nanoparticles or nanomaterials in agriculture. So, in the last class we talked about the mechanism of action of iron pyrite nanoparticle in the case of a spinach crop, and in general that is possibly the most accepted theory by virtue of which iron pyrite acts. So, just for a small recap where we finished the previous class. So, if you look at it, so this is where we showed that pyrite particles producing trace amount of hydrogen peroxide and iron in +3 and +2 state and SO4 2 minus. That is the aqueous chemistry of iron pyrite, which is essentially a Fenton like reagent. It is acting like a Fenton like reagent. Okay. So, they used Fenton like reagents, okay, and this hydrogen peroxide is involved in breaking down of those huge starch molecules, which essentially is mimicking the amylase activity, and due to this breakdown of the starch molecule, this plants or these treated plants get a better startup as compared to the plants which are not getting that fast supply of energy. This is precisely what we believe is happening out there. Okay. So, in other words, we can call FeS2 water system as an artificial starch breaking enzyme system. Okay. And apart from it, it is believed that it triggers another pathway which is yet to be explored, is called a Brassener steroid pathway, which is essentially a pathway which gets activated when plants realize that it has to acquire more nutrients in a short period of time. Maybe it is a kind of related to some way to the stress pathway. It is not very clear, but it seems that this is one of the things which is happening apart from the H2O2 signaling and of course fortifying the plant in the environment of iron and sulfur. Now, if you look at the concentration of different elements which are increasing in this plant, so if you look at it, iron, that remains constant. There is no difference between the treated and the control. Okay. So, control versus treatment is same. Now, if we look calcium, it is definitely in the treatment, seed treatment, this is higher. If you look at manganese, this is higher. So, manganese being higher does make sense because chlorophyll is higher. So, it means your manganese clusters will be higher. If you look at zinc, zinc is higher. Similarly, if you look at sulfur, sulfur is higher. So, these elements are higher in the case of the seeds which are treated with iron pyrite nanoparticles. So, these plants have this added feature in them. Now, from here, if you look at the different kind of crops where it has shown, so the field trials which were done with beetroot showed a very profound impact. In case of beetroot, we observed a much modified, or there is a modification in the root structure. Root is much more thicker. So, if you take a cross section of beetroot, then you will observe the beetroots which are treated has a much more thicker cross section as compared to the beetroot which are untreated. Okay, something like this. As you know, the beetroots are like this as compared to the one which are untreated. So, much more thicker cross section. So, it is found that there is a significantly higher beet production by nano-FES2 seed treatment, and the total yield of control and FES2 pre-treated is in the range of, in the case of control, it is approximately 4.65 kilogram, whereas in the case of FES2 treated, it is almost like 6.85 kg. Okay. Similarly, if you look at the carrots, if this is the case of beetroot, and if you look at carrots, then you observe a very interesting improvement. In the case of control, it is 11.4 kgs, whereas in the case of test, and this is all per unit area whatever we are talking about in the trial. Okay. So, there is an increase in approximately 19%, and the contrary, in the case of beetroot, you observe an increase of approximately 47% increase. Okay. So, similarly, if we go to fenugreek, which is a commonly used spice in African, Asian and Latin American continent, you will observe that the leaf area is much more bigger in case of fenugreek. So, the leaves of the fenugreeks out here are much more larger as compared to the leaves of the control. Okay. Similarly, in the case of mustard, it was observed that there is approximately 65% increase in the yield, that is. So, here now we are talking about a oil seed which is 230 grams, whereas in case of it is 380 grams. So, it is 65% increase in the mustard. Similarly, in case of sesame, if you look at it, so what we will observe is there is a higher production of sesamum, which is another oil seed, higher production, and similarly in the case of alfalfa, which is a fodder crop, there is a higher increase. So, if you look at it, the profile of crops, you will observe the beetroot, these are the roots, carrots, fenugreek, mustard, sesame, alfalfa. That is almost covering two vegetable crops. These two are the vegetables. Then, of course, some people use fenugreek also as a vegetable or as a spice. It's a spice crop. Then you have mustard, which is a oilseed crop, mustard and sesamum. And here you are having a fodder crop. So, these are the ranges of crops which have been already tried out, and it has been observed in the field trials, and this is the data we are talking from the field trials. These field trials have clearly shown that nano-FES2 application is improving the yield just by simple seed treatment. Okay. So, now if you look at this chart, you will realize that there is one set of crop which is missing here, the one which is the major chunk of our food, which is a grain crop. So, the challenge was that time to try out a grain crop, something like rice or wheat. So, next set of trial data, what I am going to show you where FES2 has been used is seed treatment of rice. Well, rice is grown in two different ways. Either it is grown by direct seed treatment, or it is grown during transplantation. So, you grow the rice crop in the nursery and then you bring them back into the field and grow them. So, let us see what is the seed treatment data of rice seed, but before I get into that, these crops what I have talked about, at least in the case of analysis of the fenugreek, the flavonoid contents what has been observed has shown that most of the flavonoids which are present, that quercetin, gallic, ascorbic, asparagine, lutein, they are more or less same. Okay, there is not much difference in their concentrations. Okay, so this is one piece of information what I wanted to share with you. Now coming to rice, so the way rice experiment was done, and well, for those of you, I will be providing all these papers for you to read. So you can download these papers and read them. That will kind of give you an idea how these kind of experiments are being done. So in the case of rice, the experiment was done slightly differently. So what we wanted to observe was, does, of course, by this time it was clear with these multiple crops that FES2 seed treatment indeed increases the production of the crop. Now the question was, does this production, how far it is from NPK treatment? So as I told you in the beginning, excess use of fertilizer is a challenge, because excess use of fertilizer led to contamination of the soil resources, water resources, and it creates a lot of other problems. This is one aspect of the story, but the other aspect of the story is, there's a large continent called Africa where affordability of fertilizer is not easy. It's a huge challenge there. Similarly, there are parts of Latin America, their affordability of fertilizer is a big challenge. And by the same token, the small and marginal farmers of Asia, Southeast Asia, can't afford fertilizer unless there is a government subsidy, and this government subsidy and availability of fertilizer is not something so easy. It's a very tricky ball game in the agri-economic center of our country. So one of the major requirements is, some way or other we have to reduce the fertilizer consumption. How one can do it? So in order to test this, so if you look at it, the major fertilizer consumption happens in the grain crops, where the big chunk of land investment goes for this growing these grains. So this experiment was done in a way, so the seeds were taken. Now we talk about the rice crop. Okay, so seeds were divided into 4 different categories, as we did in the beginning: simple water control, simple FES2 control. Okay, so the first we did it like this, FES2. Now after 12 hours of treatment, and by the way, the dose is approximately 100 microgram per ml of water. So that essentially means, suppose you have, for all the seeds whatever you are going to treat, if you have 10 ml of water for that, then you will have, that will contain 10 microgram of nano pyrite. Okay. So, post treatment, the FeS2 fragment was divided into two parts. I will tell what being done, and the water treated were divided into two parts, these seeds in equal number. Okay. Okay. Now, what was being done is, these were grown in field without just whatever residual nutrients was present in the soil. So, without supply of any NPK, whereas the other water treatment seeds were grown just normally the way you grow with NPK. Now, these two sections, FeS2 treated, these were also done the same way. Here without NPK treatment and with NPK treatment. So, you observe there are 4 different categories: simple water treatment of the seeds divided in 2 parts. One was given in a complete control condition, though no additional fertilizer whatsoever, whereas the other group get a normal fertilizer dose what is being given currently across the country or the particular regional location where this experiment was conducted. So, then the FeS2 treated seeds also underwent the same thing. One was given a control, like grown in the field where there is no added fertilizer was given, the other one was given fertilizer. So, this is the case which has the maximum input. If you look at it, this one has maximum input, both seed treatment as well as fertilizer. Okay. So, now after treatment, what was observed? There are 2-3 interesting observations which emerged out from this study, and the observations are as follows. If we look at the days to maturity for these crops, so now, and these are now in the field. So, if you look at days to maturity is for all the groups. So, if I name them as A, B, C, D. So, for all A, B, C, D, it is all more or less the same. Okay. There is no difference. Now, if we look at the plant height, so if you look at the plant height, you observe a slight increase in this group, barring aside A is equal to B is equal to C, but here D is slightly more, but not significantly. Okay. Now, if you see the effective tiller per plant, effective tiller per plant, in that case what you observe is there is a significant increase in, as compared to A, which is without NPK and just water treatment, B, C and D are higher, okay, more or less same. And in terms of the yields per kilogram, what was being observed was, in the case of control, whatever talk about this situation A, it was 1.32, okay, kilogram per hectare, whereas in the case of standard fertilizer application, it is, which is a group B, which is 1.61. In the case of NPK, only NPK, which is the C, without only FES2 without NPK, no NPK, minus NPK, and this is just plus NPK. The number was around 1.63, whereas in the case of FES2 plus NPK, the value was 1.68. So, in other words, what we are observing is that nanopyrite seed dressing in the case of rice leads to a NPK equivalent rice production. So, this is what emerged out from much of these field trials, that FES2 indeed does some very interesting change it brings about in the seed, which is manifested in the form of grain yield as well as vegetable production. So, now if you look at what all crops have been covered as of now. So, in terms of the vegetable field trials of the crops, in terms of the vegetable, spinach, then you have beetroot, then you have carrot and fenugreek as a leaf. There are several people who use fenugreek as a green leaf for dietary consumption. In terms of oil seeds, sesamum and mustard, okay, and partly sunflower. Then in terms of forage crop, alfalfa. Spices, of course, we have talked about fenugreek, and in the grain crop, it is rice. So, rice is the only one which is a monocot. So, if you look at the spectrum from dicot to monocot, it is pretty much working for the whole spectrum of grass family to legume family to Solanaceae and all other families. So, the rice trial clearly indicated now we are equipped with a tool which provides NPK equivalent reduction, which is a feat which was never achieved earlier. So, this is something exceptionally interesting to note, that it is possible that one can get an NPK equivalent system. So, in other words, biology has way too more to offer. NPK is not the only way you can get higher yield, but then what are those other ways? So, in the next class we will talk about another aspect of iron pyrite, how this potent molecule can come very handy to grow crops in nutrient deficient soil, because here you have observed that it can help in producing equivalent amount of yield as compared to NPK. Okay? Thank you.